I don't know if you could hear me, so I'm going to repeat again. So this today's, I'm trying to teach you the monohybrid and dihybrid cross. Probably I'll just teach you today a monohybrid cross. This is specially dedicated to my students in CBSE who are going to do their 10th board, and they are struggling with this concept. So like my tuition students, so I'm thinking that why not work on this and help everyone out. I hope you can see my screen. I'm not sure if it is you can see my screen here. Yes, so that's nice. I think you can see my screen. So let me explain you that what is the monohybrid cross. So this is a monohybrid cross. Why this is monohybrid cross? Because if you see, we have taken one single trait, right? We have taken one single trait, and this is for height, correct? So this is for height. Now, what exactly we are doing? You have heard of Mendel, right? He's my favorite scientist. Why he's my favorite scientist? Because he is the father of genetics. And the time he was born, nobody used to go to colleges and universities to learn, right? And so he was self taught, self motivated. So that is the one thing I admire hugely about Mendel. The second thing is that in his whole life, he was never recognized that, he's the father of genetics. It was only done when he was recognized when actually he was dead. After his death, even after 30 years or so, people realized that what a great person he was. So since then, he is my favorite scientist. So he actually explains that why we are how we are, right? So monohybrid cross is the cross between two parents taking one trait. Is the cross for one trait. Now, one trait could be height, color of the flower, because in 10th grade, you are just focusing on pea plants, color of the flower. So, one single thing we are taking, right? Or a seed type, whether it could be wrinkled or smooth, right? So, we are taking one trait at a time. That means either height or color or type of seed, like that. Got it? So these are both alleles. When we are writing TT, this is what? Homozygous dominant. All right. Why homozygous? Because you can see the both letters are the same. Correct? If I write, both letters are same. Correct? But they are small letters. So in genetics, what we call it as homozygous recessive. And the third type is TT. This is called as heterozygous. That means both alleles are different. Both alleles are same. Both alleles are same. But capital letter, so it is dominant. Small letter, so it is recessive. And this is heterozygous condition. That means one tall and one small. Tall, small. So that's that way we call it as heterozygous. Now I'm going to explain this cross now. So we are having two parents. So that is called as parent generation. When they cross, you know that what happens is that the parent generation, gametes are formed. When the gametes are formed, the alleles separate. I will repeat one more time. Listen to me carefully. When in parent generation, gametes are formed, alleles separate. Gametes are formed, alleles separate. So it becomes what? If it is TT, it will become T and T. If it is TT, it will become what? I want you to write down in your book, like along with me. T, small t, small t. So this is also a gamete. This is also a gamete. Haploid, right? One set of chromosomes. This is separate. This is separate. And if it is heterozygous, what it will be? It will be T and T. So gametes are ready now. This condition. Okay. So what do we do? When gametes are ready, what do? We cross them. Okay. So when you are crossing, you can cross like this. Independent assortment. Okay. Here is the law of independent assortment. What is the meaning of independent assortment? That this one is dominant and this one is recessive, but they will still mix with each other. It's not that the capital will, the dominant will mix with only dominant. It will mix with dominant, recessive, and also with the dominant. So there is no any kind of segregation here. Okay. There is no like exclusiveness here. The dominant trait will mix with dominant as well as the recessive. And this is called as the cross. Okay. And it follows the law of independent assortment. Some teachers, they teach you in detail. So that's why I'm teaching you. Though at this point, when you are just few weeks away from your finals, I would highly recommend to stick to the past paper questions and do as many questions possible. That will help you to do your best performance in your exam. Okay. So this is a cross. Okay. You can show like this, but I will highly recommend the Punnett square. Okay. Why I will recommend the Punnett square, I will tell you. Because Punnett square gives you clarity. Okay. It becomes very easy. So what exactly you are doing in Punnett square is that you just write down the gametes. Okay. You make just 4 boxes. Okay. Like this. You are making 4 boxes like this. I will make it separate again. So this is one thing, and it is made of four boxes, right? One, two, three, four. You will just write the gametes. Okay, capital T, small t. And you can write small t here and capital T here. There is no problem with that. Let me just write this for you like that. There is no rule. So don't think that, only capital can be written on sideways or something. No, it is not like that. Okay, you can write like that, this also. And then just write this T is going to pair up with this T. This T is again going to pair up with this capital T together. This small t is pairing up with this T. This small t is pairing with this capital T. So, right? After this, we are supposed to understand the ratio. Okay. So this was monohybrid cross. So far we have reached for a square, a cross. And this one is called as Punnett square. This method is called as Punnett square. That it was the name of the scientist. There is no other significance. It was just name of the scientist, and because he gave it to us, that's why it's called as Punnett square. Next thing I want to definitely explain another cross, okay, with another ratio also, because so far I have not explained the concept of ratio. I'll just scroll down. I really hope that you have understood so far. If you have any question and if you are watching my video right now, you can leave message. Okay, I will get back to you. And I tutor on a regular basis. I have students from different curriculum all over the world. If you are interested in taking lessons from me, you can always join me, leave a message, and I'll reach out to you. Okay, so next I'm going to teach you, like this was a cross between homozygous dominant and homozygous recessive. Now let me do between two heterozygous plants. Okay, so this is the parent generation. Now we will do the gamete formation. You know that we just have to separate that, right? If you circle it, it is little more clear, obvious to you. So that's why you can do like this. And I will straight away, this is called as F1 generation. I'll straight away go here. Familiar boxes, as you know, it is called as Punnett square. So I'm not writing it again. Okay. Now I'll write T, 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 and T. Okay. As I showed you before, the pairing will, you have to do the pairing. This goes with this. This goes with this. Do along with me so that, you can check later that if you got it right or not. Now look at this one. This is small and this is also small. Now we have different kind of combinations, right? We have one, they're homozygous dominant. These two are heterozygous, and this is one is homozygous recessive, correct? So there are two words, ratios, you might have heard of in a lot of questions, you'll find this. One is called as phenotypic ratio. And then other is genotypic ratio. What is the meaning of this? Phenotypic means how it looks like. Okay? How it appears. So whether it is this plant with capital T, this plant capital T small t, this plant capital T small t, they will all be tall, correct? Why they will all be tall? Because capital T is dominating over small t. Capital letter shows that it is a dominating over the recessive one, over T, which is recessive. So if you just count, this is 1, this is 2, and this is 3. So that means it's 3. What about this one? The small t, small t. This would be just small letters, right? That means it will be a short plant, homozygous recessive. So for the first time in all these three, this is going to express and it will be 1. Okay? Tall, 3, short, 1. Phenotypic ratio. How it appears. Now, genotypic is little more detailed than this. What is the meaning of genotypic? Genotypic means that, okay, it appears tall and it appears short. But what exactly is the genetic makeup? That means we are looking at the allele. Okay, we are looking at the allele. So, this is the first one. If you let me change the ink so that probably it will be. This is, both are dominant, right? 1. Now, 1 dominant, 1 recessive, 1 dominant, 1 recessive. So together what? 2. 1 dominant, 1 recessive, 1 dominant, 1 recessive. 2. And this is both are recessive. So again, such kind of plant is just 1. So genetic makeup wise, this is just 1. So that is called as genotypic ratio. Maybe you can try this cross. Just by changing the letter, it becomes confusing, isn't it? So you try RR into RR. Okay? And what you will do, you will form the gametes and you will do the Punnett square. And also you'll give me phenotypic and genotypic ratios. Okay. So I think that I would like to stop here because, I have explained you and I have given work also. Next class, I'm going to explain you that how exactly the dihybrid cross works. Okay. And similarly, I will give you explanation on some important topics in chemistry and also in physics because I teach all three. In case if you want lessons directly with me, this is the time you just message, reach out to me. Okay. And I will. Okay. Thank you so much for watching. Have a great day.